Lungs provide the interface between the external environment and human body. Inhalation of approximately 500 litres of air per hour brings millions of particles, such as soot, toxicants and infectious agents, into the airway epithelium. Thus, lungs are exposed to a variety of insults that might trigger molecular changes in their cells, which eventually can lead to lung diseases such as asthma and COPD. To evaluate the pathological effects of the exposure to airborne toxicants, such as pollution and cigarette smoke, inhalation toxicology studies can be conducted in animal models. One of the goals of the 21st century toxicology is the so-called 3Rs approach, which encourages researchers to reduce the number of animals they use, refine the assays to reduce distress, and ultimately replace animals with alternative methods whenever possible. Philip Morris International is developing tissue culture models and computational methods to gain mechanistic insight into the effect of exposure on different test systems. These systems can be used for mechanistic toxicology. The classical monolayer cell system is a tool that is used in a wide range of toxicology studies. However, ongoing research is demonstrating that 3D tissue cultures more closely mimic the natural environment than submerged monolayer cultures. 3D organotypic bronchial, nasal and buccal tissues grow at the air-liquid interface, making them amenable for direct aerosol exposure. In nasal organotypic cultures, the typical ultrastructures such as ciliated cells, basal cells and goblet cells can be observed. In our laboratories, to simulate what might be occurring in the lungs, smoking machines generate cigarette smoke or aerosols in a controlled and reproducible way. The organotypic tissues are then exposed to the generated smoke or aerosol in the climatized exposure module. After exposure, these tissues undergo a set of downstream analyses in order to assess the acute exposure response and biological impact. Histological analysis evaluates the effect of the treatment on the morphology of tissues and cells. The preparation of histological samples starts with fixation, which is required to preserve tissue and maintain lifelike structure. Then, the tissue is processed, embedded in paraffin wax, and cut into thin slices by a sharp knife using a microtome. Slices are placed on a glass slide and stained with dyes to give both contrast to the tissue as well as highlighting particular features of interest. This is what we see under a light microscope. Finally, digital images are captured with a slide scanner for further analysis. In parallel to the histopathological analysis, a cytotoxicity assay is performed on tissue cultures. Assessing cell membrane integrity is one of the most common ways to measure cell viability and cytotoxic effects. Some cells, when insulted by smoke or aerosol constituents, undergo necrosis or apoptosis and release cytoplasmic components into the culture medium. Once in the medium, the released components produce luminescence by reacting with a dye added by the scientist. Finally, the culture medium is harvested and the concentration of the released components is quantified by a luminometer, which determines the magnitude of the luminescent signal that is proportional to the number of dead cells in the culture after exposure. To assess functionality of lung epithelia in the exposed tissues, we measure the ciliar beating rate of the cells in the tissue culture plate. After smoke exposure, the motility of cilia on epithelial cells of the respiratory tract slows down or even stops, which eventually reduces the removal of mucus from lungs. The cilia beating movement is recorded by means of high-speed camera, and the cilia frequency rate is determined. To gain further insights into mechanisms perturbed by the exposure, whole-genome transcriptomics analysis is performed. After exposure to cigarette smoke or aerosol, organotypic tissues are collected, processed, and RNA molecules purified. The gene expression profiles are determined by the quantification of RNA molecules. One way to quantify the differentially expressed genes is to use microarrays. Once isolated, RNA molecules are prepared and introduced into a microarray where hybridization process occurs. This method allows us to quantify more than 20,000 transcripts at once, coming either from an exposed tissue or a control tissue. In addition, other methods such as quantitative PCR technology allow scientists to confirm and complement microarray measurements by targeting specific genes of interest. An alternative to quantify the differentially expressed genes is to use RNA sequencing. As its name implies, counting the number of RNA sequences for each gene is used to quantify gene expression after exposure, similarly to microarrays. In addition, RNA sequencing allows us to look at alternative spliced transcripts and mutations. RNA sequencing can also detect different RNA types, including messenger RNA and small RNAs, such as microRNAs and tRNAs. Proteomics is emerging as a complementary technology for mechanistic toxicology assessment. Thus, we apply proteomics tools for a sensitive, comprehensive, and highly quantitative detection of proteins in tissues and cells and their secretome. In particular, mass spectrometry-based proteomics is rapidly evolving through the development of new instrumentation and computational methods that allow untargeted and targeted abundant quantification of thousands of proteins in tissues extract after exposure. As a complement, proteomics targeted approaches focus on proteins of interest. The antibody-based proteomics approach supports the rapid and targeted analysis of a large number of samples, such as for the measurement of released cytokines, markers of inflammation in the basal medium. To interpret data generated by the in vitro approaches described so far, we leverage novel computational methods. For example, by describing the relationships between the mechanisms involved in the regulation of biological processes, causal biological network models offer a framework for identifying mechanisms modulated in response to active substances exposure, as well as quantifying the induced perturbations. Causal biological network models are built using the relevant scientific literature. They represent the causal nature of relationships between biological entities such as proteins, DNA, and RNA. In our laboratories, causal biological network models are used to interpret large-scale transcriptomic datasets obtained by microarrays or RNA sequencing to quantify the impact of exposure on an experimental model system. Leveraging algorithms approaches, the changes of RNA amounts in response to exposure allow us to identify which biological entities are perturbed and to which extent they respond. In this way, the effect of exposure on each biological process is modeled in the network model, such as cell fate, cell proliferation, cell stress, and inflammation, can be visualized as star plots. We use all these methods to assess our novel tobacco products. You can get more information about these methods and results at www.pmiscience.com.